Okay, shall we start? So let me just give a short 1-minute summary of what we have been discussing yesterday. So the basic take-home message is that even single-celled prokaryotes have a sense for chemicals in their environment. They can sense them and then can take decisions based on their sensing of the outer environment. And one immediate behavioral consequence of sensing the chemical environment is to take decision and swim towards sources of nutrients or away from noxious substances. And this behavior, which is called chemotaxis, we have seen this, how it is, how it expresses at the level of populations. So on the macroscopic size, we see populations drifting towards sources of attractant. We have seen that, how genetic mutations can impair this behavior, and this was the key to the discovery of chemoreceptors, specialized transmembrane proteins that detect the presence of chemicals in the environment and transduce this information inside the cell. And there will be definitely today more on this, on how this proceeds. Then we moved from the population level to the single cell level, and now we're currently trying to understand what is the physics experienced by a cell at the micron scale. And the first observation that we did is that at those scales, water looks like a very, very viscous fluid. So this is the very different physics of fluids from the one that we experience every day. And one particular effect is that fluid flow tends to be reversible because of the strong viscosity, which impedes coasting, that is, the ability to exploit inertia to move. As a result, this was the last slide yesterday, it's impossible to proceed forward in a very viscous fluid by a sequence of movements that is totally reversible. So if you had two rigid oars, and you moved that only in this way, you can't move. You just oscillate back and forth forever. You can never progress with a steady drift towards the direction you wish. Slightly larger organisms like algae, which have flexible flagella, Chlamydomonas is one example. For instance, it swims pretty much in a way where there is an upsending part like this, then the flagella go outward, and then pushes like this. And this sort of motion is not reversible, see, making a cycle. So this allows the algae to proceed upward in that specific case. So here I'm showing just to warm up to very short movies that sort of give a visual analogic demonstration of this principle. In this case, there is a swimmer. Actually, here there's a battery, and here there's a flap that just goes back and forth. So according to this scallop theorem, that is, reversible movements don't produce net drift, this swimmer should proceed. The fluid here is, I guess, corn syrup or some oil, highly viscous fluid. This is real time. So you see it's left here. I'm marking the head of the swimmer. It swims, basically goes nowhere. After a while, but that's just because it's not perfectly unbalanced, so it tends to go upward like this. But it's not progressing, you see, in neither direction. It's only changing slightly its angular momentum. Now, what happens if you use, sure. This is, sorry?
What is controlling whether it's reversible or irreversible?
Various parameters, so the size, the speed, and the viscosity. These three ensemble make for the Reynolds number, which is a non-dimensional number. So it's speed times dimensions divided by viscosity. If this number is very small, everything will be reversible. If it's very large, it's going to be like ourselves moving in air, and then it's going to be irreversible, basically. And here, the speed you see, it's very slow. It's, I don't know, it makes 1 flap per second. The size of this whole object, you've seen the hand, so it's sort of 10 centimeters, 20 centimeters. And the viscosity of the corn syrup is much, much larger than space-time velocity, which makes for a reversible movement. Now, the setup is the same in the next slide. What is changing is that now to this battery have been attached 2 coils, 2 rigid coils. The simple fact that these are coils, they're going to turn, will make the motion non-reversible, because a coil, either it turns in one direction or the other, but it's mirror symmetry, right? So this simple spatial arrangement breaks the symmetry of forward and backward motion, and then should produce a net displacement. And this is basically the intuition on how flagella work to propel bacteria. So I'm again marking the starting point here. You see it's proceeding very slowly, but definitely going to the right. So this is sort of pedagogical illustration of how this reversibility can be broken by a spatial arrangement without introducing flexible flagella, which is another way. I mean, Chlamydomonas uses flexible flagella to make 2 different motions. But if your flagella are rigid, you cannot do this sort of motion. And then you have to rely on some other mechanism, and helical flagella is one of them. Not the only one, but an effective one. So very shortly on the physics of how can helical flagella provide a thrust, that is, physically propel the bacterium. Actually, the theory is very, very old, and it's pretty simple. The basic idea is that you can grasp it from very, very simple experiment. So imagine that I have this object. This is a very slender object. When I immerse it in a fluid, it will offer resistance to motion. The resistance to motion when it falls like this, it's larger than when it falls like that, right? This is just because of the geometry of the object. Now, what happens if you let this fall slantwise? So you keep it slant and you let it fall in a very viscous fluid. Of course, in air, if I do it, it's another thing. It's another Reynolds number. I won't say anything. But in a very, very thick viscous fluid, where would it go? Would it fall like this? It would fall like this, right? It will fall slantwise because the resistance which is offered to motion in this direction is very large. It's less in this way, so it will fall slantwise. Now, that's the only physics you need to know to understand how this thing works. Flagella offer a resistance which is smaller along their direction and larger in the other. So as a result, you just write down the diagram for the forces in the system. And if you just drag your helical flagellum in this way, it will exert a force in that way. And that propels the flagellum, right? So it takes just 2 minutes to write down the vector force balance, and it's just force, effective force proportional to velocity. But the 2 components have 2 different coefficients because the drag is different in the 2 directions. And that's all you need to know. Very, very simple. So let's keep it with this very small setup of physics information. We move back to E. coli. How are the flagella organized on its surface? And now it comes something which is first sight sort of surprising, because the flagella are attached at random places on the cell. Other flagella have polar arrangements after bacteria. So Chromatium ocani has a bundle of 40 flagella at the pole. But E. coli has somewhere on the surface, no particular point of attachment for them. On average, there are seven flagella, but they can be as small as 3 or 1, or can be up to 10. No particular constraint on this. And all these flagella are actually, we'll see that in a second, attached to the surface via a hook, and then there is a rotary motor, which we'll see in detail in the following, that makes it turn. So when all these motors turn in one given direction, which is conventionally said the counterclockwise direction, all these flagella coordinate, and they move in the same direction. Okay, so there is behind the cell body there is this bundle of flagella all together, and they propel consistently the bacterium in the forward direction. So you can see here a movie taken from Hauerberg's lab. Here, the flagella have been made fluorescent. So what you're seeing is the fluorescence emitted by flagella. No, sorry. And you can see them streaming, right? Now what happens? That from time to time, given the concentration of some molecules in the interior of the cell, that we will see in a second, one or more of the motors may decide to turn the other way. And when this happens, well, that's pretty critical, because the good functioning of this bundle is related to the fact that, you see, all these flagella are just entwined one with the other. If all the motors rotate in the right direction, there is a wave that propagates from the cell body out. But if you make them turn in the other way around, this wave will come toward the cell body. And at a certain point, it would just exert a force where the hook is, where they are anchored. And in fact, when this happens, when there is at least one of these flagella which is turned in the opposite direction, there will be a retrograde wave, and then one of these flagella will just be expelled from the bundle. Well, just by mechanical forces. And as a result, when this happens, the bacterium feels an angular momentum and changes direction. And this event is called a tumble. It may occur because 1, 2, or more flagella at a time or at close times change directional rotation. And here you can see many of these bacteria performing these irregular changes of direction due to this change of change of sensor rotation of the motors. You see, the bundle comes apart here and here and here, and the motion becomes highly irregular. As a result, at the behavioral level, when you look at the motion of bacteria, you track them, their trajectories. See, this is one bacterial trajectory seen from 3 different directions, from the top, from the side, and from the front, the same trajectory. And the behavior that you see, in absence of any specific chemical cue, okay, so it's a perfectly homogeneous environment. The bacteria don't know where's the north or the south, no information about that. And you see that the motion is made of straight runs. This corresponds to the situation where all the flagella turn in the same direction. And these are interspersed by rapid changes of direction, which are the tumbles, where 1 or more flagella are expelled from the bundle. The duration times of these two phases are very different. So runs last on average 1 second, order of magnitude-wise. These tumbles are faster events. They last 1/10 of a second, typically. The duration of these phases is not set deterministically, but rather follows approximately a Poisson distribution. These are exponentially distributed. It's a random motility process. So there will be long runs and short runs. On average, they will all last about 1 second, but that's just the average. And for tumbles as well, there is an exponential distribution, which is a much smaller duration. You might wonder whether there is a specific selected direction of outcome of the tumbles. When I just change direction, what will be the distribution of angles? Actually, it turns out that it's pretty broad. It may even sometimes occur that bacteria just make a 180 degrees turn. But there's a slightly favored angle in the forward direction. So the average angle of turn is actually not 90 degrees, the one that you would have for a completely random rearrangement of direction. It's slightly bent forward. Another important thing that one has to notice in this data is that straight trajectories are not that straight, actually. So the bundle is turning in the same direction. It's continuously fueled. But yet, you see that they wander around. So what's the reason for that? Now you must realize that if you are an object which is 1 micron sized, the environment that you see around you is something which is very, very agitated. Thermal agitation produces water molecules hitting you all the time, and this is something which is far from being negligible. And actually, the fact that these trajectories are not quite straight is because there is thermal rotational diffusivity. Okay? So the effect of these water molecules hitting on you produces a relatively small effect at the level of displacement when compared to the swimming velocity. Okay? But it produces a quite important effect. Sure.
One flagella among the full bundle, if it starts rotating in the other direction, in that case also it may produce such diffusion, right?
Yeah, right.
So can you identify it is because of the
It is because of internal signaling that it changes rotation.
Can you identify whether it's because of thermal signal or whether it's because of one flagella is rotating in the other direction?
No, thermal fluctuations cannot induce a change in rotation. They're not energetic enough. You cannot switch the flagellum in another direction because of thermal fluctuations. It's just energetic budget. I mean, if you just compute the energy that it takes to do that, the set of and the KT energy that you can allow by thermal fluctuations is not sufficient. So when you just estimate how much this 1 micron size object is sent off course because of the hits that it receives with water molecules, you see that this information is quantitatively encoded in the rotational diffusion coefficient, which tells you how many degrees squared per second it will go off course. And if you put the figures, then you clearly see that roughly after 10 seconds of straight motion, okay, so suppose I produce a mutant that is not able to tumble, which is of course possible to do. It will tend to go straight, but then after 10 seconds, it will have completely lost memory of its initial direction because of water molecules hitting on him. So this 10 seconds time scale sets a very important time scale in the physiology of the swimming bacterium, because in a sense it's a time scale which you don't want to go. You don't want to have responses over time scales much larger than this, because you will forget what your opacity is in direction-wise in any case. So this sort of sets an upper limit to the ability of the flagellum to keep track of the memory of its previous motion and its previous sensing on the environment. So it's very important to keep this in mind. There is this typical time scales of 10 seconds which you have to deal with. So this is sort of typical motion for a bacterium. This sort of swimming is basically on all the time. It continuously consumes energy. And the only situations where the bacterium just sits and stops is whether either nutrients are so low that bacterium starts considering the opportunity to turn into a completely different state of mind and metabolism, and maybe going towards sporulation for some bacteria. So it's a completely different switch to another behavior, another lifestyle. Or if nutrients are so large that actually it's not worth spending all this energy for moving. It's costly, but it's not that costly actually to swim. If you make a budget, energy budget, it's still a relatively small fraction of the energy that you spend for your own metabolism and for building up the biomass to double. Okay? So it's expensive, but it's not so energy consuming. So you must really go very, very low in nutrients in order to see this behavior to stop. Okay, so that's basically what happens when you see them swimming. Now, if you just look at these trajectories seen from a distance, then they basically look like random motion, right? These very short parts, you don't see them anymore, and you look at trajectory with a very low spatial resolution and time resolution, then what you see is just a sort of three-dimensional Brownian motion effective one. And if you try to measure the diffusion coefficient for this effective Brownian motion, you can estimate basically from the persistence length of the walk and the typical velocity that it has. So the typical diffusion coefficient of a bacterium, effective, is basically its velocity squared times the duration of a run. It's dimensional estimate, the simplest one that you can cook up, and it's pretty good. And then if you put the figures, you get this number, which is orders of magnitude larger than the effective diffusion coefficient, than the actual thermal diffusion coefficient, right? That's just because it's moving a lot with respect to what hits with what the molecules can provide. So default motion of a bacterium seen from a distance is this alternance of run and tumble, which produces a sort of random displacement in space. So this is basically what we had to know about the motility properties. So how it swims, how it moves, which is, of course, a necessary condition for chemotaxis. And in fact, evolution also reflects this fact, because if you go and look at the how is the genome and the transcription machinery organized in E. coli, you discover that the genes for swimming are expressed before the genes for chemotaxis. So you maybe know that in E. coli the genes are organized in operons, so in functional units that couple several genes, and then these operons often are switched on sequentially. So I start the transcription of one operon, which will produce proteins associated to a set of genes, and when this transcription ends, it will start transcription of other genes, only causally, one before the other. And so all these genes that make up the motor, the flagella, are expressed before the genes for chemotaxis start, which makes perfect sense, because in order to perform chemotaxis, you have to swim, etc. So that was about the mechanical environment that a bacterium sees. But now we'll move to something which is even more interesting, which is the chemical environment that the bacterium sees, which are the physical-chemical constraints of what is the olfactory perception of the bacterium around itself. And this is very important because we'll set up the conditions to understanding how bacteria perform foraging. That is, how they look for nutrients, and they exploit them. So a very similar argument to the one that we did for the Reynolds number applies also for the diffusion of chemicals. So how is in general described the diffusion of a chemical in a fluid? It's described by this equation, which is the advection diffusion equation. It contains two terms. This is the diffusion part, molecules of water hitting onto your favorite chemical and displacing it into the fluid with a molecular diffusion coefficient, D, which is related to, proportional to the temperature of the fluid, inversely proportional to the effective friction coefficient of that molecule in the fluid that you can relate to the gyration radius through some appropriate microphysics hypothesis. And this part instead is the coherent transport by the flow, which displaces the concentration accordingly. So the balance between these two terms, so how you're transported by the flow and how you diffuse into it because of thermal fluctuations, is given by a non-dimensional number, which is very similar to the Reynolds number. It's velocity, the size, but instead of being divided by the kinematic viscosity coefficient, it's divided by the diffusion coefficient, okay, which has the same dimensions, meters squared divided by seconds. And now if you plug the typical values for the bacterium, which swims at about 30 microns per second, which, by the way, it's pretty fast. It's of the order of, say, 15 body lengths per second, which for humans would mean that we could be able to run typically and so in a sustained manner at roughly 50 kilometers per hour. This is the length, 1 micron, typical length. And this is the diffusion coefficient of a typical molecule like an amino acid or a sugar in water. And if you put all these things together, then you see that this number is small. And this number is small means that this term is dominant with respect to this. So even if bacteria swim like crazy, around them the environment is dominated by diffusion. Which basically means that you don't get much by swimming just, I mean, it's not like bacteria are like, I don't know, flying with their mouths open in order to catch the insects in the air. It's not that analogy at all. So the basic flux of nutrient to the cell surface comes from diffusion. And this is very important because it leads to very, very different dependence of the intake, for instance, on the size of the bacterium. So if you were to catch nutrients just by, if this term dominates, okay, so there are nutrients here, and I'm moving very fast, and I'm catching them like this. They are attached to them. And then the amount of nutrients that I get, the flux of nutrient to my surface, is proportional to my surface, right? But if diffusion wins, it's not like that. It's proportional, it's linear in the size. This is a consequence of the fact that diffusion is very regular motion, okay? And molecules are continually moving away. So even if I make a near miss and a molecule misses me, it's very likely to get me again. And that makes it for a linear dependence rather than a quadratic dependence, which is much larger when you're small. It's much better to have a linear dependence rather than a quadratic dependence, right? Of course, it depends then on the parameters, but the parameters are this. So actually, saying that this quantity is small is equivalent to say that the flux that comes to your surface because of diffusion is much larger than the additional value that you might take by swimming. So swimming is not made for getting more food, right? So it's just, if you stand still, you basically get the same, roughly. So motion is not made for increasing the flux, but it's made for some other purpose. So now let's try to get some physical insight of what this very, very viscous and diffusive environment, what constraints it puts on the ability of the bacterium to sense concentration, to measure them, and then to take decisions according to this sensing. So the first question is, the environment, as we said, is dominated by thermal agitation. So the first question that you should ask is, can bacteria measure concentrations reliably? So suppose the concentration is very low. So there are few molecules of nutrient around you. So they continuously come, hit the surface, maybe hit a receptor, maybe not, then go away. So the signal that you might see, it's actually maybe very noisy. That basically depends on how many molecules hit the surface at a given time. So can we estimate what's the typical precision of this measurement that the bacterium do? It's very, very low-level arguments. Huh? Yeah. The flux is the number of particles unit time, right. This is the concentration that is far away from the cell, and here we are assuming that the cell is able to absorb everything and I will comment on this assumption shorter on. And this is the size, right? So basically, if the process is diffusive, this is the only dimensional combination that you can come up to produce a flux 1 over time. And the 4 pi is the fact that you have a perfectly absorbing sphere. This is known as the Smoluchowski formula for the flux to a surface. And it's actually very simple to derivation of this. You just assume that everything depends on diffusion, and you have a static profile, so you kill everything. And then you solve for this equation, Laplacian of concentration equal to 0. And for a sphere, there is a radially symmetric solution, which says that concentration actually will depend as a constant plus 1 over r. And then you match these conditions to the surface of the sphere and outside, and then you get the flux. There's nothing much to... Actually, this is also for those of you who have a background in physics, this is absolutely equivalent to the Gauss theorem in electrostatics. For those who don't have this information, forget it. But the basic idea is that there is a constant flux due to diffusion throughout all scales, and that's the value that it takes at the surface of the sphere. So first question, can they measure reliably? Also, we will go with a very simple estimate, actually. For those who wish to have a thorough reading, there is this beautiful, beautiful paper, 1977, by Berger and Parcel, whose name is Physics of Chemoreception. That's a classic, very easily and enjoyable reading. And I'm just taking some excerpts from that paper to give you an idea of back-of-the-envelope estimates for how bacteria can effectively sense their environment. So the flux, as we said, according to this formula, is 4 pi d diffusion coefficient, size, concentration. Okay. So the total number of molecules that will hit the surface in a given time, allotted time, capital T, is just flux times time. And that's it, right? So how can we estimate the error that we make in measuring the number of molecules that hit the surface? Okay. We just assume that, as it always done, if this number is sufficiently large, not too large, but sufficiently large, the error will be typically the inverse of the square root of this number, right? Central limit theorem, usual stuff. Independent hits of molecules on the surface. Not very refined argument, but that is sensible. So your signal-to-noise ratio, right, is basically this number. This tells you how precise you are in your measurement of the number of molecules that hit the surface. And this is a proxy for the concentration. Then you put the figures, and you put a time of the order of 1 second, right? So that's the typical time for one run. So the question you're asking is, during the time that bacterium makes one single run, is it able to evaluate reasonably the concentration that is around it? Then you just plug in the figures for a typical concentration where the bacterium responds significantly to this. You know that this is something which is consistent with behavior, with actual behavior. It's not the smallest value that can be actually appreciated by a bacterium, but it's on the lower end of concentrations. And then you see that basically you have a precision that is less than, sorry, you have an error which is less than 1%. So this is a very precise measurement, actually. So 1 second is enough, given this concentration, to make a reliable estimate of what your environment is in terms of concentration. There are no specific physical limits at this concentration. When you go around 1 nanomolar, 3 orders of magnitude below, then it starts to be tricky. You can always compensate with longer times in that case. But that is, if you have a very noisy environment, the simplest way to remove this noise is to average out. You make several independent measurements. After all, the average will emerge from your statistics. But it will take time. And as we said, it's not obvious that the bacterium can allot all this time. So at a certain time, there will be a concentration where measurement will become difficult below a time scale of the order of 10 seconds, which is the one that you want to keep track of before losing your direction, right? But at this concentration, there's no problem of precision. Concentration can be evaluated precisely.
So you are assuming that each particle is effective, so that there are enough receptors.
Next slide. Of course, the assumption here is that it's a perfectly absorbing sphere. Okay, that clearly cannot be. It would mean that all the surface is filled with receptors, and all these receptors has to be for the same chemical species, and then I could not respond for it. No, that doesn't work. Doesn't work. Now, the nice thing comes again that the basic motion is diffusion. So what matters again is not the surface of the cell which is occupied by receptors. It doesn't depend on how finally you cover your surface with receptors. It doesn't depend like the ratio of the areas, but it depends like the ratio of the sizes. Again, because of diffusion, which means suppose you have your sphere with your cell surface, the surface of the cell, and then you just put some receptors onto it. You do not cover it completely. What will happen to a chemical that arrives, and typically it doesn't hit the receptor at first sight. First hit, it just will be on the surface of the cell. And then typically either it binds non-specifically and it's released shortly after, or it completely just rebounds on the surface. The nice thing of diffusion is that it doesn't flow away. It stays there. It hits the surface many, 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 many times continuously. Tan, 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 tan. And then diffuses around, and then it searches, and finally it hits a receptor, and it's detected. So as a result, the fraction of area that you have to cover with the receptors in order to get, say, 50% of the total flux is very small. It's a very simple calculation that you can do. So here is the picture. There is the cell with the receptors on the surface and all the signaling molecules that are impinging. This is the average profile of concentration, as discussed before. Then you just compute analytically what is the flux to the receptors, which is you take a sphere, some parts of the spheres will be absorbing, some others will be reflecting. It's a problem of the theory of diffusion, not particularly simple, not particularly difficult, but you can work it out. And you see that the incoming flux is the incoming flux for a surface which is completely filled with receptors, and then there is this prefactor, which is always less than 1, as it should be. And crucial dependence is on this number, which is number of receptors times the size of one receptor, but it's not the surface of one receptor, it's the size because of diffusion, and this is the size of the cell. So as a result, this number of receptors that you can allocate would scale like a surface, and this would scale like a size. So that's very nice, because just by covering less than 1% of the area with receptors, which are nanometer-sized objects, you can allocate with 1,000 receptors with this area covering, and you get 1/2 of the full flux. So you lose 50% in the flux, factor 2, not too bad, but you have saved 99.99% of the area. And so you can allocate every sort of receptors there, receptors for aspartate, for serine, for whatever. You can allocate everything. There's room for everybody. And for all of them, okay, I will just sense half of the molecules, which is not a big expense, after all. Okay? That's the power of diffusion. Diffusion, of course, it's also a nuisance. Because if in your external environment, suppose you have a patch of nutrient, okay? So some algae has died. You're a marine bacterium. An algae has died and has released all its cytoplasm, which is rich in very interesting stuff for a bacterium. And then you want to go there and exploit this. But diffusion will very quickly dissipate away stuff. diffuses by definition. It goes very fast away. So you must be able to be there before this cloud of nutrients vanishes, right? So the distance that you travel in a given time must be larger than the distance by which this cloud of nutrients has diffused in space. Is it possible that bacteria can do that? Of course, this sets a limit on velocity, typically, given the size, or on the size given the velocity. Let's put the figures for bacteria. So the condition is that you can get there fast enough before this thing vanishes. So this is the typical length scale that in a time t, this cloud of nutrients will have formed. And this is the distance that you can travel, right? If this distance were much larger than this, you just would have seen this diffuse away without the ability of chasing it. Of course, this sets a condition. Then again, you replace the very same figures that you had before, and then you get that the typical velocity that enables bacteria to chase diffusing nutrients is 30 microns per second, which is order of magnitude-wise exactly the one that bacteria have. There are much faster bacteria, but it's very untypical that you find bacteria that swim at less than 10 to 20 microns per second individually, which makes sense in this fluid environment because that's basically, given the size and the velocity, that's its limit. Okay. Next step. It's very good to measure concentration, but we have to measure concentration differences in order to know where the grass is greener, where there are better prairies, where there's more food. We must make differences. So the first question is, do bacteria, such as E. coli, measure differences in some way, say, taking differences from the head to the tail? Right? If there's a gradient, the head will see, suppose the head is this way, whatever it means for a bacterium, the head will see more concentration than this, then the bacterium may have some way to compute differences between head to tail, and then decide to proceed in that direction by some way that we now will discover what it is soon. Is it possible? Is it feasible? So let's try to make, again, a little estimate. So the criterion is that the difference between head and tail must be larger than the noise, than the fluctuations. So given a certain concentration, you estimate how many molecules typically, on average, you have in the front and bottom, and if it's too small, noise will overcome your signal and you won't be able to tell front to back. Now, here we set ourselves to the lowest level of detection of gradients for bacteria, which is particularly impressive, at a given concentration environment, which is of the millimolar. So actually, if you put bacteria in a millimolar environment and you subject them to changes which are as small as relative gradients of 10 to the minus 6, which is very, very small gradients, you can still detect responses. They can bias their movement towards the upper side of the gradient. That's sort of very, very lower limit of detection. And here we put the condition, that is, this epsilon, which is the relative gradient, that is the percentage difference in concentration between head to tail, which is written like this. So it's the size times the relative gradient, which is a number. And this number must be larger than number of molecules inverse square of the number of molecules. That's the level of noise, right? So signal larger than noise. This is a constraint on the measuring time, right? Because, again, if you measure long enough, that will be okay. Just enough time is enough to get an arbitrary precision. So the time must be larger than, you just unwrap this formula, replace all the numbers, and then you get 100 seconds. Well, that's way too much. That's way too much to take decisions effectively on the time scale of a single run. That's because rotational diffusion imposes a limit to the way you can really change the behavior behavior in response to the environment, which is 10 seconds. So it doesn't work this way. It must be said that this is true for E. coli, but other bacteria, larger ones, may measure differences in space. There are bacteria which are vibrants, marine vibrants, actually they live in seawater, which are longer, and they actually measure concentration differences across their span, which is roughly, say, 5 to 10 microns. So it's a factor 10, which appears here, which brings this time scale to something similar to their way of motion. They also perform longer runs. It's a different motility, but they perform longer runs, so they allow for more time for decision. Okay, so it's trade-off between all these different quantities, how long you perform a coherent motion. This sets a limit on how long you can measure, etc. If you're larger, rotational diffusion is smaller. If you're larger, you can have ability to measure difference in space. So it's not a general rule that bacteria can't measure gradients in space. E. coli just can't, given its behavior and its physical properties. Then how does it do? Well, the answer is actually pretty simple. It measures gradients in time. It has a time memory. So rather than making difference in space, it just keeps a time memory. What were the concentration that I measured 1 second ago? What's the concentration now? I make the difference. And since I am moving, this difference in time translates to difference in space, or vice versa. So time information is converted into space information. So let's estimate. Can this work? If we make a very similar estimate, the only difference is now that we are evaluating the relative gradient in time. So that's how the concentration changes in time, because I am moving over a time t. And if I plug these numbers, you see that with respect to the previous expression, there is a factor, velocity times time divided by size, that appears in front, which multiplies this term. And actually, it is this prefactor that makes the little miracle. But this is just because if you run, actually, you might think that the bacterium is making comparison between the point where it starts and the point where it arrives. So it's not anymore across 1 micron, its size. It's across 30 microns. It's the size of one run. And this enables to make sensible measurements. And then the constraint over time becomes, okay, it has to be larger than a fraction of a second. And we are back again to reasonable timescales. Okay? These are, of course, plausibility arguments. What is striking is these were done in 1977, and how actually bacterium E. coli measures concentration, which is in time, was experimentally proven years later. Okay? So it was, to me, it's a beautiful example of where simple theory can give reasonable insight on biological behavior. It's all very simple, right? So let's go to the experimental test of this hypothesis.
Good morning, everybody. We have gathered here today to mourn the death of Maya. He was a skilled helper in our department. He passed away on 15th December. Mr. P. Lakshmaya was born in 1981 as an helper in the Works and Maintenance Department. He was transferred to Department of Biochemistry in the year 2001. And he was the lead.
Prove that actually bacteria sense the concentration difference in time rather than in space. Okay? Actually, it's a very ingenious experiment. The idea is that you should submit bacteria to a uniform concentration which changes in time rather than exposing them to a gradient, and to see whether their response changes. So your environment is uniform in space. There's no preferred direction, but it changes in time. if bacteria are able to sense time differences, they should respond to this. If they are able to sense only space difference, they wouldn't respond, right? Of course, the idea is very simple, but the way out you measure the responses may be very tricky, because at that time it was not obvious how you could track all bacteria and then measure their response, their difference. It was not that obvious. So they came up with a very ingenious solution. What you do basically is you take bacteria and you take off their flagella. Yeah? You don't need to strap them out one at a time, of course. Just some mechanical strain in a pipette is sufficient. So that you reduce your population of bacteria to bacteria that typically on average have one single flagellum. Then you prepare a glass and you cover this glass with an antibody for flagellin, which is the protein that makes flagella. And when you do that, what happens is that the bacteria get attached to the surface with the flagellum, right? So those bacteria that have no flagella will just float around and disappear. Those who have two or more flagella, all of them will be stuck at the surface. And those with a single flagellum will have one flagellum remaining and the cell body outside. But then when the motors turn, it means that since the flagellum is tethered to the surface, it's the cell body that will rotate, just to preserve angular momentum. So you can see the cell body rotate rather than looking at the flagella, which are very small and difficult to see. And if there are two flagella attached, just the bacterium won't move because it's totally tethered and it can't rotate around that. So that's a picture taken with this tethering assay, and you can clearly see bacteria cell bodies rotating in one direction or the other, and then from time to time changing direction, and you can see bacteria which are totally still, which are probably the remnants of those which have more than one flagellum attached to the surface. And then you can put a stimulus, a uniform stimulus, remove it, and play around and get the response to this. And if you see a response, that means that they measure concentration in time. But more than that, you can actually quantify this response. You can make a very decisive step towards quantitative understanding of the behavioral response to chemical stimuli. Actually, what you can do, you can characterize an object which is very keen to physicists. You can measure the response function of bacteria. That is how the behavior changes as a result of an impulse stimulation. So you just give a very, very localized in time stimulation. So no concentration of aspartate. Concentration of aspartate very high for a short time, millisecond, then no concentration. So for physicists, just stimulating the system with the delta function. You measure the actual, what is called the response function. This is one of the fortunate cases where biologists and physicists call the same thing with the same name, the response function for both, right? So if you do that, what will be your response function? In this case, will be just the fraction of bacteria or the fraction of time that is spent running into one specific direction. If they turn counterclockwise, this is a proxy for the actual behavior of bacteria running. And if they turn in the other direction, it's a proxy for bacteria tumbling. So it's a this has direct behavioral impact on measurement with direct behavioral interpretation. So you do this experiment. It's in the 80s, 86. So this is the bias ratio of bacteria turning into one direction to the other. This quantity, of course, goes from 0 to 1. This is bacteria that tumble all the time, and this is bacteria that run all the time. So behaviorally, this corresponds to a bacterium that goes nowhere, continuously changes direction, and this is a bacterium that tries to go as smoothly as possible, modulo rotational diffusion. Here there is no stimulus, and you see that there is this level of response. Then you provide this very short stimulus, very localized in time. So the attractant is there, and then they're not there anymore. As a result, after less than a second, the fraction of bacteria that turn into one direction, that is the direction of running, increases rapidly. And then it falls down. It falls below the original baseline, which is very striking. So in response to a positive stimulus, they will tend to run, but after a while they will tend to retreat from this and to make more tumbly behavior. And then after 5 seconds, basically everything gets back to the baseline. So the memory of the bacterium of that stimulus ends at about 5 seconds after the stimulus. Of course, very large stimuli can induce longer effects. So the memory depends also on the stimulus. If you give them, just flood them with the attractant, this curve will just stay up there for a very, 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 very long time and then will fall down. But then if the stimulus is small enough, you go into what is called in physics the linear response regime. Then the amplitude would be basically proportional to the amplitude of stimulus. And the response time should settle to one definite uniquely defined time, which is of the order of 4 seconds. This is sort of the minimal memory time of the bacterium, 4 to 5 seconds, which is, again, of the same order timescales of run and tumbles. Everything is there, orders of seconds. Everything must take at that time scale, less than 10 seconds, of the order of 1 run, maybe 1 to 5 runs, order of magnitude-wise. Okay, so if such thing as a linear response exists, we should be able to prove that bacteria do respond linearly, actually, in a range of concentrations. And how you test that? Rather than subjecting bacteria to a short pulse of attractant, you give a more common stimulation in biology, just give a step stimulation. So no concentration, then you raise your level to, say, 10 micromolar aspartate, and you leave it there. And then you observe the response to this. If linear theory applies, the response to a step, you could derive it from the response to a pulse. Just because a step is actually a superposition of many pulses, just one side the other. So the response function, which is this curve, must be related to the response function to a step. And actually, technically, it's just one is the convolution of the other in time. It's just the response to a step should be the integral over time of many of these responses, just one after the other. And then you do the experiment, and you measure the step response, which is this wiggly curve, and you compare it with the response obtained by the convolution of this single impulse response. So the smooth curve is an outcome of the first experiment. And the wiggly curve is another independent experiment. And these two work, which means that there is a range of concentrations where the response is linear. Of course, then you can try different concentration, etc., check all this. I'm summarizing. It works. There is a definitely consistent range of stimuli where the response is nearly linear, which is extremely good news for theory, by the way. There's another emerging result from this experiment, that when you submit bacteria to a step response, to a step stimulus, the response after 5 seconds gets back to the original level. Now, this phenomenon is actually well known to us all. It is sensory adaptation. You change the environment, you respond transiently, but after a while you get habituated to the environment and you don't respond anymore. And your parameters get to the usual value. So a small bacterium such as E. coli is capable of this sensory response. This is something which we experience every day, right? So now we were in a rather dark room, shady. We get out in the sun, and then it would take some seconds for us to adjust our vision to the new environment, and then to be able to detect contrast again, right? So it seems that something similar is happening here. So conventional explanation of sensory adaptation is this one. It's a purely sensory one. It's something that tells you you move from one environment to another. You must be able to compute differences, must be able to detect contrast. So you want to move your range of detections. You have to have a dynamic range of sensitivity that accompanies the stimuli. right? And so this seems to be the case. Actually, this view has been recently challenged, and we discussed in what sense it might not be the right explanation for this sensory adaptation behavior. But on the face of it, it looks like pretty much like the response of sensory cells to vision or to hearing. So the shortest time scale is of the order of a few seconds. Again, if you put a very strong stimulus, it will go up, and then it can stay there for, say, even 15 minutes before recovering. It depends on the property of the bacterium. Yes, it does. It does. Actually, it can change. In isogenic bacteria, it may change from one bacteria to another, actually. They might have different timescales for response. They may vary actually by a factor of 10. It's something which is regulated epigenetically. But we will delve into the molecular biology in a second, so I hope this can help understanding all these details. But this is sort of an average behavior. It's not individual at the time. Okay, very good. So the response is linear and adaptive, sensorially adaptive, on a short time scale, the memory of bacteria. At the level of trajectory, so bacteria in the wild, now what happens is that they use this time differencing algorithm in order to make run longer when they just go in the right direction. So if they are swimming and the concentration is increasing, this response tells that they are sort of taking differences, and if the gradient is positive, if the concentration is increasing in the right direction, they will increase the duration of a run. And if it's decreasing, they will shorten the duration of a run, and this will produce a bias in this random motion, which will, on average, propel bacteria towards higher concentration zones, areas. So that's how this individual response is connected to the drift of bacteria towards higher concentration. So you might wonder, how is this realized? How is this response realized at the molecular level? So let's delve into the hardware of chemosensing and chemotaxis. How do bacteria do that? And it's particularly enlightening because it's one of the simplest transduction systems, and it's one of the systems that we understand the best and we can model with the most of details and the best of understanding of the parameters, kinetic parameters that appear in this model. Actually, it's sort of textbook example of a signaling pathway. So let's spend some time in it and look at the actors of this signaling. So this is a schematic representation of the cell. These are the membranes, right? So here is depicted only the inner membrane. E. coli has two membranes, actually, an inner and outer one, and gram-negative bacterium. So these red sticks are the receptors, those famous receptors that were whose existence was inferred by Adler. In the periplasmic space, these receptors bind either the ligand or other binding proteins that themselves bind the ligand. So it might be one-step or two-step process. When the ligand binds, as in other receptors, this changes the conformation, the spatial conformation of this receptor, and this affects into the behavior of the cell into the cytoplasmic space. So this is the way this transmembrane protein transduces the signal from the outside to the inside. So what happens on the inside? These receptors are linked via scaffold protein, KW. It's essential to chemotaxis, but it just plays a role of a scaffold protein. So it just makes the connection between the receptor and this more important protein, which is a kinase. It's a sort of bizarre kinase in biology because it's a histidine kinase, whereas typically in eukaryotic biology we have serine and threonine kinases all the way, and tyrosine kinases. But it's very uncommon to find a histidine kinase, but that's what it is, sort of a remnant of all stages of evolution, but it works perfectly as a kinase. And this kinase typically is able to autophosphorylate itself. That is, it can attach itself phosphate groups. So this phosphorylation mechanism is one of the key signaling mechanisms. So it's one way to say that a molecule is on or off, basically. And they transfer these phosphate groups from kinases to others, and there are phosphatases which take away this phosphate group. So signaling relay from bacteria to higher eukaryotes heavily depends on this phosphorylation mechanism. These are covalent modification of proteins. So the protein is there, sequence is given, folding is given. The only thing is that new pieces are attached, small groups, which are very important. Phosphorylation, methylation, ubiquination, all these covalent modifications are playing an enormously important role in establishing the behavior of cells, the actual behavior. So this protein tends to be self-phosphorylated. So in absence of ligand, there is a certain fraction of these proteins that will have this phosphate group attached, which sort of lights on. It's switched on when it's kinase. This kinase is switched on. Upon binding of attractants, it happens that the level of phosphorylation actually goes down. So it works sort of the other way around, which is often a sort of confusion, but never mind. What is important is that the state of phosphorylation of this protein changes in response to ligand binding, that is to the external environment. This state of phosphorylation is actually relayed to another molecule, because eventually we need to put ourselves into communication with the motor. So this signal has to affect the motor behavior. If there is a attractant, the motor will have to change direction of motion. So we must convey this signal from outside to inside receptor, but then from the receptor to the motor. So we need some protein that carries the message inside the cytoplasm. And this small protein is this KY, which can act as an acceptor of this phosphate group and relay it to the motor. So basically it's a shuttle that goes there, takes the phosphate group, diffuses in the cytoplasm, and then takes it to the motor. And when this KYP binds to specific proteins of the flagellar motor, then in that event, the motor gets the signal to change direction, to tumble. Actually, phosphate transfer from KY to the motor is the signal for tumbling. So when an attractant comes, the level of phosphorylation goes down, the number of these shuttle molecules phosphorylated goes down, and the signal for tumbling goes down, which means that you go straight. Right? Everything is in the negative, but the effective result of attractant is that you go straight. So this is the sort of forward signaling from the signal to the motor. But there's something that must account for sensory adaptation, right? Because if you had just this part of the signaling pathway and you give a positive stimulus, this will, and you leave it constant, just like a step, this will continuously affect the state of the motor. So your response function would be something that goes up and stays there. It doesn't get back. It doesn't adapt. You need a molecular mechanism for adaptation. Now, this is provided by another sort of covalent modification, which is methylation. So that the attachment of methyl groups now to the receptor. This procedure of attachment of methyl groups, what it does, it just sort of resensitizes the receptor and brings the level of phosphorylation of Ka back up, resetting the system to 0. It's sort of system reset. It is also coupled to the signaling via this feedback loop, which provides, is understood as this way, additional robustness to the system, but is not necessary. You could cut this feedback loop and let these Kb molecules act. Sorry, you could cut this part of the loop here, and then these molecules would adapt as well, but in a less robust way. So pretty much is known about this, the kinetics and the, all these networks. You basically know all the rate constants for these reactions. So this covalent modification is which is methylation, which is operated by two enzymes. One that methylates, which is methyltransferase, which is here, and one that demethylates, which does the opposite. This is this key B. This pair of enzymes is crucial. And actually, it is this reaction that sets the longer timescales of the memory. So this process of methylation is the one that resets the memory. So it's the one that takes the longest timescale. And so depending on how many molecules, given the rates, depending on how many molecules of Kb and Kr, you will have a different response in time. And now the thing is that how many molecules of this kind are there into a bacterium? About 100. And since there's 100 molecules, you can clearly see that from one individual to the other, one might have, say, 80 molecules, the other one 120, and their timescales of response will be different. Their behavior will be different. These are individual changes which are completely phenotypical. 100 per cell. Yeah. Thousands. So it pretty much the behavior depends on, it's limited by the number of this. So the rates are importantly limited by the number of molecules that you have at your disposal. And then there have been experiments when these two proteins have been overexpressed, and then you see that the individuality disappears. They all behave the same way because the individual fluctuations in the number of molecules that you have disappears. So it's very important because this is a very generic mechanism for individuality in single-celled organism. Just epigenetic individuality, which might also be inherited from one generation to the other, because when you divide, you give copies of your proteins to the cells. Since these proteins are relatively stable, you can inherit the phenotypic state, sort of Lamarck evolution at the level of single cells over a few generations. Because then proteins are degraded, are newly synthesized. So there is, of course, a change in this. But still, it's very, very interesting and important as a basic mechanism in such a simple organism. It's pretty fast. Actually, it's in order of milliseconds. Because the diffusion coefficient is 1,000, basically, micron square per second. And the distance is 1 micron. So basically, it's sort of instantaneous. There is a very slight gradient that establishes from receptors to motors. But since they are not particularly arranged spatially, you don't really see inhomogeneities inside the cytoplasm in the distribution of these molecules. So it's pretty fast and homogenized by diffusion. Now, theoretically, what is interesting is that this system actually can be described with a very, very compact set of equations. Right? Now, for a moment, forget about the real kinetics when there are numbers of molecules that fluctuate up and down, and then take a very, very simple description in terms of concentrations of objects. Then the basic take-home message is that you just need a dynamic system with 2 degrees of freedom, 2 variables, 2 scalar variables, to describe the dynamics of the system. So as a dynamical system, this is something which is very, very simple description. The configuration of the system is just given by 2 state variables. One is the level of methylation. So what's the fraction, average fraction of sites, of methylation sites that are occupied by methyl groups, right? This M. And this Y is the fraction of active messenger proteins inside the cytoplasm. All the rest is basically fixed because the binding, unbinding, and autophosphorylation reactions are very fast, so they are basically in equilibrium. So there's equilibrium between the ligand and the receptor, between the receptor and the phosphorylation state. So all these reactions actually give you an equation rather than giving you a differential equation. So these things are instantaneously equilibrating, basically, because the time scale for this is order of the millisecond measured. The same thing happens at the motor, very fast reactions of binding, unbinding at the motor and response, chemical response. So basically, the frequency of counterclockwise rotation is a function of the fraction of messenger proteins that, that are present in the cytoplasm, active messenger proteins. So we will discuss a little bit about where this formula comes from, because it's very important. Actually, it's an example of an allosteric cluster. And we will discuss a little bit cooperativity at the level of the motor. This is the part that competes to methylation. It's important to understand adaptation. And this is perhaps just the most innocuous part of the equation, is just how the signal is relayed from receptors to the motor. But then three important things happen here and here. That is amplification of the signal here and here, adaptation of here, okay, by a different mechanism, which are very important. Of course, you might ask, okay, that's very well, good and nice, but suppose I'm fed up with the E. coli and I want to switch to B. subtilis, another bacterium. So what have I learned after all? Should I start everything back again and it's completely different stuff? Well, the answer is no, because basically all bacteria and archaea share the same architecture of signaling of chemicals. Actually, this is the general model of how signaling works in bacteria and archaea. This class of receptors, methyl-accepting receptors, are widespread. They come in different flavors, but they're basically emerging from gene duplication and mutation mechanism. So they are copies of the same original template for a receptor, which has been used and reused again. And again, you have the same sort of key A molecules, the same shuttles. There are additional molecules coming in from other species. There are this one in green or yellow, whatever, right? But when you look at these molecules, actually you see that these are just structural and sequential copies of other proteins, just duplications and slight deletions, additions, right? So they all very obviously come from a very reduced set of proteins that have been exploited. You know, evolution is a tinkerer. It uses things that are there, and if it's possible to use them again with another purpose, then that's pretty much a good idea. Okay? So what we learned for E. coli is basically good intuition for what happens in other organisms at the molecular level. Bacteria and archaea. Eukaryotes, it's a different story at the molecular level. So these receptors come actually, as I said, in different flavors, which respond to different stimuli. So there are receptors for serine, threonine, and other amino acids, receptors for aspartate and maltose, so they mix amino acids and sugars, via special binding proteins, right? So there are always, or nearly always, intermediates that mediate the interaction between the receptor and the ligand, so that they can connect always to the same docking site, but different stimuli. And then other dipeptides and pyrimidines, sugars, and receptors for oxygen, etc., and many other things. And you might be interested to know, knowing that E. coli, of course, responds also to temperature. It has a favorite temperature, which, no surprise, it's 37 degrees, the temperature of our gut. And can you see any sensor for temperature here? No. Just because they use these two receptors together to sense temperature as well. So they're just abusing the function of these two receptors in order to measure temperature differences. So all these sort of signals actually, like a funnel, get into the same pathway, which is the one that I was describing before. So this also sets very interesting questions, not all of them answered, as to, in presence of several different stimuli, how can the bacterium tell which is which? If, after all, everything goes into the same memory, which is this fraction of phosphorylated KY proteins. And it's not obvious. Yeah, that's what I was saying. For now, everything has been mostly focusing on how it responds to individual stimuli. But this idea of multiplexing the signal onto the same line is something that still has to be explored. So it's an open question, how we can deal with multiple. We know behaviorally, we know what it does. We can expose them to different signals, contrasting signals, and we know that it makes choices. It prefers to go in one direction or the other. It's not confused by this. It just takes consistent decisions. But how this is disentangled and what other information might be necessary, this is an open question. Okay, so very quickly, now we will go through some detail of this organization of sensing, which actually gives us some very interesting general lesson about how you respond to stimuli, what are the ingredients, and what are the benefits, etc. So one very important thing is that for chemotaxis to be effective, there must be at some time an amplification of the gradient. So you want to be able, you compete for nutrients, you want to be able to detect very small gradients in order to be there fast, reliably, soon, get the food before others come, right? So there is a strong pressure towards sensitivity. You want to have very exquisite sensitivity to gradients. And in order to do that, you need to amplify your signal because it's very weak, and then you would turn out into consistent behavior. And in fact, that's what bacteria do. Indeed, by FRET experiments, which is a technique to visualize proteins binding inside a cell, it's been possible to relate the fractional change in receptor occupancy, so how many molecules, ligand molecules, are bound to the receptor, to the change in the kinase activity, that is the intracellular response to stimuli. Okay? So this is something which tells you the input-output response just in the first part, the sensing part, before actuation to the motors. So it's the top part of the signaling. Ligand converts into kinase activity inside the cell. So these are the experimental data in under various conditions and various stimuli, all step stimuli in this case. So that's the very fast response at short times. And then when you plot them onto a linear scale close to, that's the linear regime, you see there is a proportionality between stimulus and response. So the upper curve and the lower curve are just by increasing or decreasing the stimulus, so stimuli in the positive and in the negative direction. And you see that's the fact that this is linear in this range of changes of fractional occupancy means that there is a linear response regime. The fractional change in receptor occupancy is a measure of how many ligands you have in your environment. And the constant of proportionality that is here is 35, which means that 1% change in fractional occupancy of receptors. So you change from, say, 50% receptors occupied to 51%, will translate in the behavior changing by 50%. Inside the cell, it means that the level of active kinases doubles. It's enormous amplification already at this step. How is this accomplished molecularly? Well, that's a very, very general lesson. You can get very, very large amplification by clustering receptors together. It's a form of cooperative signaling. Yeah. Can you speak up because I can't hear you?
I don't really understand how you read this graph, because if you look at the big one and you go to, so you go to a half, it doesn't look linear in that.
Yeah, that part is, so first of all, this is log scale.
Okay.
Right? So it's difficult to detect a linear behavior on a lean log scale.
Okay.
This part is definitely non-linear in any scale you put it, because these are very strong stimuli. You're just going from 0 to millimolar concentration, then you go from 0 occupation to 50% occupation. So that part is certainly non-linear. The part which is linear is here, but you don't see this because this is log lean, so this is actually, these are logarithms in this plot.
It's logarithm. Okay, thank you.
Sure. So thanks for calling and for pointing out. So this is an enlargement of this region in linear scale, and that's where you see the linear behavior. Sorry for being confusing. So the trick is that receptors come in clusters, closely packed together, and actually, when they come close packed together, they form these hexagonal arrays, which are widespread among all bacteria. So these receptors actually typically pack in this form. They form these sort of arrays. And these arrays are not only a geometrical arrangement of receptors in space, but they are also functional, which means that close by receptors talk to each other. And what happens is that altogether they can change from active to inactive in a concerted manner. So it's not every receptor is by itself and it binds and then it inactivates or it unbinds and deactivates, but it's rather groups of receptors that altogether decide to switch or not to switch. And this is sort of actually the simplest example of cooperative allosteric response. And as a result, there is amplification. So I will go very briefly through the very two basic models for receptor cooperativity which are relevant for this example. So these are very, very simple ideas actually. So you start with a model where you have a single receptor which can switch from an active state to an inactive state depending on binding and depending on methylation. That's just the conceptual scheme of the pathway that I was showing before for a single receptor. Then all these receptors, you know, that are arranged close together. In the simple two-state model, it's just a collection of these independent entities, each by itself. This model by itself does not provide any degree of amplification. It's just each receptor works by itself. It's basically depending linearly, the hyperbolic dependence of the response on stimulus, typical ligand binding kinetics, nothing really can happen this way. But then you start putting interaction between nearby receptors, which means that if one receptor basically is active, then the probability that the nearby receptor is active also larger, increases. So there are nearby interactions. So this is very well known in physics. And it basically is like having an Ising model with site energies, which depend on the ligand binding or unbinding. And then you have coupling between nearby sites, which favors the formation of clusters of receptors with the same activity levels, right? So there are basically two situations. So one where these groups of spins, or receptors if you wish, are coupled together very intensely. So this J function is very large. So they do behave as a single object. So all receptors come active or inactive at the same time. This is the celebrated allosteric model by Monod, Wyman, and Changé, which is a very basic mechanism for allosteric response, or response of a protein as a whole to stimulate with several binding sites. And actually, this is the one that is relevant for receptors clusters. So receptor clusters behave as a single unit, so they are strongly coupled together. So they switch from active to inactive as a whole. But then there is also a possibility that this J is order unity in some sense, so that actually what happens is that if there is a bunch of receptors that are active, this activity will spread to nearby receptors, but they won't flip altogether up and down. They would just tend to induce close by receptors to flip, but that would be a softer mechanism. And what is interesting is that this model is good for the motor as a cooperative macromolecular assembly. So for those of you who don't come from physics, actually, this the description of the allosteric model, which I'm giving here, so it's this energy, but the spins sigma high can only be 0 or 1 for all of them. Actually, as a very simple the eraser. As a very simple biochemical description, which might be more obvious to you. And actually, this was the way that it was historically introduced by Monod and co-workers. So you may have your protein or your receptor cluster in two possible states, either inactive or active. And it can have this cluster of receptors, or this protein may have no ligand bound, or it may have one ligand bound, or it may have all possible binding sites occupied by the ligands. And then you have switching rates, say K prime plus and K prime minus, which are the same for all this. These do not depend on the number of ligands, right? But what depends on the ligand is the fact that you can bind or unbind. So this rate is just K plus times the concentration of the ligand, in excess of ligand, or K minus in this way. And again, this. And then there are also additional factors appearing, because there are actually capital N ways in which this can occur, because there are N free sites where you can put your ligand. But this occurs just with a factor 1, because there's only, if you have N minus 1 sites occupied, right? So you go on and go forth, and then at any given step inside, you have prefactors such as N minus N, K plus L, and N, K plus L, connecting between IN minus 1 and IN, right? So you have all these sets of reactions, binding, unbinding, accounting for the free sites, and then switching as a whole in an active or inactive state, or relaxed or tense state in the original description of the model. And then if you work out the equilibrium for these systems of equilibrium reactions, then you get exactly the same description here. It's exactly the same thing. It's not a different thing. Two different languages, exactly the same object. And this model was very important because actually it was the first explanation of the behavior of hemoglobin. As a result, the response for these clusters takes this form. So the fraction of active, the activity of the clusters, which is the fraction of active molecules, no matter how many binding molecules they have, as a function of the concentration of ligand. It has not a hyperbolic shape, just as binding unbinding reaction with a single receptor, but it has a sigmoidal shape like this, which means that there is a range of values where you strongly amplify your signal. Small changes in the ligand turn out in large changes in the response. So that's a basic mechanism for amplification. This works as well for the conformational coupling model, except that this happens also with a significant space coordination of the signal, whereas allosteric clusters are just holes. They act as a whole. So flagellar motor, very briefly. I just want to finish 2 slides on this, and then this is the right point to just take 1 minute, if you don't mind. So the flagellar motor, as I said, is this particular macromolecular assembly, which is rather astonishing when you see how it works. At a more general cultural and philosophical level, it's an important example of macromolecular assembly because it's one of the key examples used by people who think about intelligent design. So not evolution by itself has produced things, but some intelligent design has produced such objects. And actually, it is important to know at the cultural level that actually this object is again a result of evolution tinkering, because it's nothing but a formerly existing excretory system, very simple one, which has been hijacked to produce flagella. So it's nothing that has come up out of nowhere in evolution, but it's just been built up from formerly existing structures. The basics are that from the cytoplasm part, these KY molecules bind to this ring and then transduce a signal that forces this rotor to change direction. And the way it happens, in recent experiments has been shown that it happens, this cooperativity happens essentially by conformational spread. So the response is very steep from the concentration of KY phosphorylated to the motor response is very steep. This is one, because it amplifies cooperatively. But when you look at how this thing behaves, you can observe that there are this sort of failed attempts at switching that are not allowed in the allosteric model, because the allosteric model can only switch between two states, either active or inactive. This sort of failed attempts do not exist in the allosteric model. And actually, you can characterize the statistics of this and show that it's consistent with the conformational spread model. So the motor responds pretty much like an Ising model with this coupling between nearby molecules in activation and amplifies further the signal. So to that 35 values, actually, we should add another 10. And it already gets to 350. And then in the next lecture, we see that there's an additional factor 10. So that eventually you get to the amplification factor of 10 to the 3, which is enormous. Thank you. Sorry for being late.